I'm Kay Evanovich. Pleased to meet you all. We're going to go ahead and try and get on with this tree identification chapter 2 section, and then we'll come back to Rob. So here we go without further ado. So tree identification, you basically have the tree, twig, bud, and leaf samples that you use to identify trees. In this course, we're going to go over that, plus plant classifications, scientific names for trees and how they're written, the use of plant characteristics in tree identification, leaf arrangement and morphology, leaf shapes and characteristics, and finally, bud and twig characteristics. And I will say, as we get going, wherever you see something in your book outlined or emboldened or in italics, I would really make note of that, because those kind of things tend to pop up on tests later on, just an FYI. And if you haven't been doing the sample test questions and the worksheets at the back of your book, please start before you take the test. It really, really does help. So, and for Rob and I, usually if we repeat something or you see it repeated in the course, that might be something to take note of too. So let's get going. All right, so with tree identification, you really need to identify what tree you're dealing with before you prescribe care or treat the tree. You know, certain trees can handle things differently than other trees. You give too much water to a tree that's, not used to having too much water, you can drown them, so you really want to make sure that you know the tree you're dealing with before you try and diagnose and treat them. Diseases and pests, different requirements from, like I said, rain and all that. Species handle that differently too. So like one pest that affects a certain type of tree really isn't that much of a problem in another type of tree. But if you don't know what your tree types are, then you're going to have a problem trying to figure out what's going on. And you may diagnose something as being in bad shape that really isn't. Perfect example, catalpa. A catalpa tree has catalpa worms, and frequently the worms eat all the leaves off the trees. Well, if you didn't know that the catalpa comes back after that, it will put new leaves out as long as it's getting enough water and nutrient. This is something the trees learn to adapt to. So if you didn't know that and you saw this horrible pest that defoliated all the tree, you might accidentally say, oh, the tree is going to die because all the leaves are gone. Well, that would be a wrong diagnosis. So very important to know what type tree you're dealing with before you start trying to treat it. And your ability to ID trees really requires knowledge that you gain over a period of time. The more you look at trees and woody landscape plants at different times of the year, because they're going to appear differently in winter, obviously, especially if it's deciduous and drops its leaf, than it does during the summer when it has full leaf out, or even during the spring. Even age can make a difference on trees. Young trees tend to put out much bigger leaves. I wonder why that is. Rob will probably go over some of that in biology, but a young tree has to fight underneath the canopy of others, so it tends to put out a much larger leaf. And that can really throw off diagnosis if you don't know your tree species or the different life cycles of trees. So taxonomy, it's plant classification. And that's what taxonomy means. It's how we classify plants, right? And there's a order to this thing. It's based on biological characteristics. And it basically goes, think back to, school. You learned about your kingdom and your division or phylum. Well, kingdom is animal or plant. It's one or the other, right? And then you have phylum. Your book is going to call it phylum, but division also means the same thing. And under that phylum, you have vascular and non-vascular types of plants. Below that, you have a subdivision of vascular, under which they can fall into angiosperms or gymnosperms. What are those? Well, we'll go over that in a minute. Underneath that, you have classes of those angiosperms, and they can break out further into or dicotyledons and monocotyledons, right? Underneath that, it becomes an order. Now, you're further defining your family, which means that family of trees has similar flowers and fruits, right? So similar flowers and fruits in the families. And then under the genus, you have very similar plants, especially in their reproductive structures. So their flower structures and their reproductive forms are all going to be extremely similar, right? And then underneath that, you finally get down to the species level. Well, you say, okay, that's a lot to remember. Yeah, it is, but there's an easy way to remember it, right? So if you think, how this flows, it's going to be kings play chess on fat gray stumps. So kings, kingdom, play, phylum, chess, class, yeah. On would be the order, F would be the family, good. G would be the genus, and S, of course, is species. So that keeps it in your head as to how they go, how they line up. Kings play chess on fat gray stumps. I like that one. All right. So let's get into some classification with kingdoms. Like I said, the kingdom for trees is plant, obviously. Its phylum or division is going to be non-vascular or vascular. So it's one or the other. Let's talk about vascular plants. They're basically plants with a xylem and a phloem. When you talk about biology, you'll realize that trees, deciduous trees especially, with leaves that fall off in the fall and winter, are going to be trees with xylem and phloem, right? That's how most trees work. Sperms are plants that have seeds covered by an ovary or fruit, okay? Flowering plants, most deciduous trees, most broadleafed evergreens fall under the angiosperm, okay? It's going to have that seed that's been covered by an ovary or fruit. Think fruit trees or nut trees, nut bearing trees, that kind of thing. And then there are gymnosperms. Those are plants with naked seeds. Did she say naked? Yes, I did. So gymnosperms have naked seeds. They're cone bearing plants, right? They're not encapsulated in a fruit or nut, right? It's got a cone like a ginkgo and a conifer. Those are gymnosperms. So just remember that difference. What's the difference between the angiosperm and the gymnosperm, right? Those may come up on a test. You never know. Okay, so the phylum. Underneath phylum, we have gymnosperms. So we're talking about those conifers like juniper, cedar, and pine. And then we're talking about angiosperms, flowering plants. Most broadleaf tree species are angiosperms. Now, under that angiosperm class, you have Eudocotyledons, right? Eudocots means two seed leaves, right? So di, if we think back, di means two, right? So a Eudocotyledon has two leaf seeds. Most broadleaf species fall in this category, and some common trees, most common trees, as a matter of fact, except for conifers, bananas, and palms, right? So a palm is never a Eudocotyledon, right? They're a monocot, one seed leaf, mono, one, pretty easy to remember. So grasses, lilies, orchids, palms, those kind of things are monocotyledons. They have their vascular tissue in bundles. And it's really interesting because a palm is basically the biggest grass in the world. Can you imagine having a mow palm tree? No, but that's what it mostly resembles, right? It mostly is more common with a grass than it is actually a broadleaf tree. So just an interesting fun fact. All right, so classes. Under the classes, we have orders. And under the orders, we have families. Remember, under families, it's plants with the same, have common characteristics, same types of flowers and fruits. And then under genus, plants that are very closely related with similar characteristics, especially their reproductive structures, okay? And then under species, that's really the level that identifies the plant. The species name is a combination of the genus name and the specific epithet or name. Remember that. See there? It's underlined. Specific epithet equals name, right? And that's how we create our tree name that we use all over the world. So the flowering structures are going to be the same on a genus, right? And then it may look a little different, but the way the reproductive structures are very similar, right, in that same genus. So let's keep that in mind. Plant classification. So here we go. This is a silver maple, and how that silver maple, which is a common name, is identified. Its kingdom is plant. Its phylum is an angiosperm, right? It has two seed leaves. So its class is a dicotyledon or Eudocotyledon. And you need to remember that. Eudocotyledon is the same thing as dicotyledon, okay? That's in your new book, the new chapter, the new 4th edition of the study guide. It is changed from dicotyledon to eudocotyledon, but it means the same thing, 2 leaf seeds. And then its order is Sapindales. All right. Its family is Aceracea, sorry. Its genus is Acer, and its species is Saccharinium. So that's the silver maple, is the genus and the species name. So it's an Acer Saccharinium. And notice how they're written. Acer is capitalized. Species, Saccharinium, is not. It's just written normally. And that's how it goes in plant classification. All right, so nomenclature, the naming of plants, remember? Common names, let's face it, they can be confusing and very misleading because one tree may have several different names depending on where you are. You know, you've got Carpinius carolinea, American hornbeam, but it's also called blue beech. Out west, they call it an ironwood. In parts of the north, they call it a musclewood. So, but it's always our Carpinius carolinea. That is an American hornbeam, right? Or blue beech or ironwood or muzzlewood, but it's always a Carpinius carolinea. Hope that makes sense. A tulip tree, that's the common name. Hey, it's a tulip tree. Well, which tree is it? It could be a Magnolia exfoliandra. It could be a Lyrodendron tulipifera, right? It could be a tulip tree or it could be a magnolia tulip, right? So tulip tree, well, that's kind of generic. It could fall into several different trees. There's Douglas fir, which really is not a fir at all. It's in a different family, right? Bald cypress, well, that's not a cypress tree. Mountain ash, hey, it's not an ash, but that's what we call it, right? So all these common names can get confusing. So remember, common names should not be capitalized unless they include a proper name, and they should never be italicized, right? So if it's a river birch, you just write river birch, just like it's written there. Cultivar names, however, now that's a little different. If you say Betula nigra, which is river birch, that's its species and genus, but it has cully in quotation marks. That cully variety can't be trademarked, right? But a common name, well, that can be trademarked, like heritage with your little trademark symbol, river birch. Whoever created that heritage by, you know, mixing trees and breeding trees, genes and splicing and all that wonderful stuff that we do came up with heritage, breeds true every time. So they copywrote the name heritage for their cully, right? Their Betula nigra cully is trademarked as a heritage river birch. Trademark names are never written using single quotations like you see in cully with the single quotes. When you're doing a trademark name, it's written as Betula nigra heritage capitalized, but not in quotations. You might want to remember that. Botanical or scientific names. So botanical names, each plant has a unique botanical name. It's the same throughout the world, and it's based on species classification systems. So sugar maple is an Acer saccharum, and it's an Acer saccharum in every country. G'day, mate. Have you seen that Acer saccharum? Well, he may call it something else in Australia in his common tree name, but he's calling it Acer saccharum just like we do here, right? Same thing anywhere over the world. It's going to be an Acer saccharum. Okay, that keeps us all knowing what the tree is all over the place. Now there are hybrids. Hybrids is a result of crossbreeding between two different species, but they're usually from the same genus. And it's written with an X between the genus and a specific epithet. Like this one is an Acer X Freemani Jeffers red. So it's a cross between an Acer and a Freemani, right? And Jeffers red is the, you know, like the cultivar name. So that's how those are written. Okay. So a species, a subspecies within a species is always marked as SSP. So your subspecies, if it is one, you're going to see that SSP, period. It's a naturally occurring, close related group within a species found in the same geographic range, but with distinctly different characteristics. Like a black maple is a subspecies of sugar maple. So it's very similar, but there's enough of a difference that it's a subspecies, right? So form is another one, and you have a plural of form, which is forma, Might want to take note of that. And that's written with a small f, right? It's similar to the subspecies, but less distinct differences, like a red flowering dogwood, right? That would be its form. Varieties are written out as VAR, period, just like it's shown on the slide. It's a subdivision of the species that has a trait distinctly different from the other plants within that species, and it naturally breeds true. It does not involve man's getting into that. It naturally breeds true to whatever that trait is. That's a variety, VAR. And then finally, you have cultivars, like little gem magnolia, right? The little quotation mark, right? Cultivar varieties, those require human intervention, asexual propagation to reproduce a trait. So you're basically dealing with a genetically identical clone. We have created clones of our trees, and that's where these come from. So it doesn't breed true like a variety. It's something that we have to create. It's man-made, right? Little gem would be a cultivar. All right, so, okay. Common names should not be capitalized, remember that, unless they include a proper name like European, that's in quotation marks, and they should not be italicized, right? Just going over all this might look familiar if you've got the chapter 4 edition. All right, botanical names written with either underlined or italicized text, and italics is always preferred. And these are some examples of properly written botanical and common names. So you have Stromia indica, which is a crepe myrtle. Okay, that's its common name, is crepe myrtle. Washingtonia robustia is a Mexican fan palm. That's its common name. Acer saccharum, we went over this, it's a sugar maple, right? And you see the way it's written. Magnolia grandiflora, beautiful, southern magnolia, love them. And then Eucalyptus globulus, right? Blue gem eucalyptus, that would be its common name. So I hope everybody gets that as to how we write out those botanical names versus the common name. All right, so principles of woody plant ID, basically it's based on morphology, the size, the shape, the appearance of plant parts. What do you look for to identify these trees? Well, your leaves and the form, the way the tree grows. That's very useful because different trees grow different ways, right? Botanical classifications are based on reproductive structures, right? Flowers and fruit, things like that. But you can use form, bark, buds, twigs, leaves, flowers, fruit, even the smell to identify and ID trees. And it's interesting, around here we get young tulip poplars, and to me, whenever you break off a little piece of a young tulip poplar, it smells like pepper. And it's the only one that does that. That's how I can tell when they're little what it is, right? There's other things that smells come into play. Sweet gum leaf, if you're not familiar with a sweet gum and you crush that leaf up, if it doesn't have the little sticky balls that everybody knows them for, if it's the time of the year when it's not producing, you can crush that sweet gum leaf and it smells like a sweet mint almost. And that's where the name, sweet gum came from. So all these things help you to identify trees. All right, so important tree characteristics. Obviously, we have the form. We talked about that, how it grows. The branching habit, does it grow straight out? Is it kind of a rounded? Do the branches themselves twist as they grow? There are certain trees that branches do that. Leaves, what do the leaves look like? Not just in shape, like, how they shape like a maple leaf or something, but what do the leaf margins looks like? Or the leaf base, or its apex, or is it hairy? Some of them are hairy underneath, some of them aren't, some are smooth. You have buds, the little place where the next set of leaves, the next branch is going to pop out. That always looks different on different types of trees. The twigs, how they branch out is different. The pith inside that twig is different in different trees. The bark is different. Flowers, fruit, may have them, may not, depends on what type tree it is, right? It helps to know all these things, and it really helps to identify them, especially in winter when you don't have leaves. You really want to know what the bark, the form, the twigs and the pith and the buds look like. It's gonna tell you what type tree that is when there's not leaves present. Now, at a glance, when you're trying to figure out what a tree is, you're looking for form. You're looking for the branching characteristics and the growth habit of how that tree grows. Is it tall and skinny as a columnar. You know, if it's columnar, it's not going to be an oak, right? Maybe there are now varieties of oaks that are columnar. So that may help, may not. But that's where you start. How does it grow? Is it rounded? Is it lollipop shaped? Does it look more like a Christmas tree shape? You know, that all helps to identify the tree. Elms are usually kind of vase shaped, you know, and then sugar maples have a brilliant fall color, very, very distinct. Live oak, oh my gosh, that beautiful spreading, dropping branches that just kind of spreads everywhere. And it may have Spanish moss on it, depending on where you are. If you're up here in the Piedmont region of Georgia, you're not going to see that Spanish moss. And yes, we have live oaks up here. But on the coasts, you're going to probably see Spanish moss draped all over those beautiful live oaks. So remember, live oaks will have Spanish moss, depending on where they are. Okay. So there's that beautiful spreading form we were talking about with the oaks. So you have two kind of forms in trees. There's excurrent on the slide that shows you it kind of, you know, grows out vertical and has a tendency to run straight up and down on the trunk, right? So the decurrent may be broken up and have several splitting major scaffold branches that go off, creating this kind of curved flowing decurrent trunk. And a way I can keep that in my head, at least when I was starting out as an arborist, trying to remember all these forms, was excurrent. That I think about a lightning strike on this tree. Isn't that terrible? But that's how I remembered it. Current, right? Like electrical current would hit the tree at the top and run straight down to the bottom. That's excurrent. Well, the decurrent would have to wander around. It wouldn't go straight down. So excurrent is straight down and decurrent would have to meander, right? Just a way to help you try and remember. All right, leaf arrangement. Leaf is determined by the presence of a bud where it attaches to its stem. On a simple leaf, you have a single, one part, one blade, one needle, that leaf that is not subdivided into little leaflets, right? A compound leaf is two or more leaflets, but only a single bud or cluster of buds at the base of that petiole. And this is the difficult thing about running this class in front of the other class, because you'll talk more about those petioles and all the pieces on those trees a little bit more in biology. But we'll go on and we will explain that a little bit further in depth on, I think, one of these next couple slides. Anyway, you have different arrangements of those leaves on the twigs. You can have an opposite where they're coming off of that little branch or twig on the same spot. See how they're attached? It's like they're coming off the twiglet at the same point, right? And then you can have alternate, which means they're coming off at different angles on the tree, different points on that twig, right? So you have opposite and then you have alternate. So they're alternating where they come off the branch. On a simple leaf, it's going to be one leaf, right? Like this silver maple that they're showing. It's one silver maple leaf. It attaches by that petiole to the twig. On a compound leaf arrangement, on the bottom where it says compound, you're looking at, looks like a black walnut. And that has many leaflets that come off, right? So it's compound. It's not just one leaf that comes off the twig. It's many leaflets attached to that twig on its petiole and petiole. And we'll get onto that in a second. So for leaf and bud arrangement, there are only a certain number of opposite leaves, right? And that would be maples, ash, dogwood, and horse chestnuts. Those are the 5. And you can remember them very simple by thinking mad horse. It's a mad horse, look out. Ah. Anyway, maple, ash, dogwood, mad, and then horse chestnut, mad horse. Alternate is basically all the other species. There are a few that aren't, but that's pretty much it. All right, so opposite is going to be mad horse, maple, ash, dogwood, horse chestnut, and alternate is going to be most other species. So here's a look at those mad horses with opposite leaf arrangement. So you have a maple up here and then your ash tree to the right of that, your dogwood with the beautiful red berries, and then the horse chestnut over at the bottom right. That's the 4, right, that fall into that mad horse category. They're opposite leaf arrangements. Okay. Conifer needle arrangements, well, that's a little different. So conifer, remember, is cone-bearing trees. Pines or pinus have needles arranged in clusters of 2, 3, or 5. That weird 2, 3, or 5. What a weird number. Anyway, yeah, so they're going to come off at the very top. Like you see, there's going to be individual needles that come off in a cluster, right, of 2, 3, or 5. The spruce, Paideia or fir, Abies, produce needles singularly, right? So they got short, sharp, single, and square needles, right? That's going to be a spruce or a fir. They don't have their needles in clusters like the pines do. They come right off the twig and they're usually very sharp and sticky. Think Christmas tree. Yeah. Other conifers may have an awl-like or scale-like foliage. This example on the bottom is a thugia, right? So they kind of look like they're stuck together in little awls or little scales that go on top of each other. So that's how you can kind of remember them. So let's get into a little bit of leaf morphology. Let's look at a simple leaf. So we have the apex is the very top of the leaf. The margin runs along the side of the leaf, right? The blade is that entire side of the leaf, right? Either side, it has 2 sides on this one with a vein running up the center. And then the petiole is the piece that attaches to the twig, right? This is a populus deltoides, right? A poplar. And then you have the base of the leaf. That can be different too. That shape on the base where it attaches to the petiole can be different for each leaf. So that's kind of the morphology for a simple leaf. And I would just kind of study this in your guides so that you remember which part of the leaf is what, where's the blade, where's the petiole, where's the base, where's the apex. This is a good one to tell you where they are. And remember, the margins on the sides can be different too. You can have serrated, not serrated, but we'll get into that in just a second. So I hope everybody follows along with how this leaf is laid out. All right, so let's talk about that margin. So you can have an entire leaf margin or a serrated leaf margin. You can have a cerulate margin, and it's showing you each one of these, how they look. And it's really different. Leaves can be really easy ideas if it's leaf season on a tree. If you got a lobed leaf, you are not going to be looking at, like a holly, because it's, not going to have that lobe. It's going to have more of a serrate or a coarse serrate or a double serrate. It's going to be sticky and pointy. It's not going to have that nice lobe shape. But just familiarize yourself with the different ways those margins can look, right? They can undulate, they can be lobed. There's all kinds of different forms of those. And it helps in tree ID. The leaf base, like I talk about, that can be different too. Remember how that silver maple looked? If you look in this diagram, it's kind of a cordate, right? Truncate, cordate on the silver maple, the way on the poplar, sorry, my bad, that last slide, that poplar, the way it kind of curved in and the petiole came down from it was kind of cordated. So you can have it acute, it can be rounded, it can be oblique, culate. There's a bunch of different ways that leaf base can look, and each tree is going to be different how its leaf base looks, right? So that just helps to further your tree ID. The shape of the leaf helps to ID the tree, right? So if you have a linear skinny leaf on a broadleaf tree, well, you might be looking at a willow oak. It's got that skinny linear leaf. Other broadleaf trees may have oval leaves or oblong or cordate or deltoid, right? It just depends what the shape is. These are all the different shapes that a broadleaf tree can have. On the conifers, you're kind of limited, right? The conifers really have four styles. So they can be scale-like, like that thugia. They can be awl-like. Think of a little awl or needle. They can be linear, right? You've got some like the Dawn Redwood, I think, has a linear needle for a conifer. And it is also a deciduous conifer. So it does drop its leaves. That reminds me of another one in why a tree ID is important. When you see these Dawn Redwoods, if you haven't seen one before, look it up. It looks like an evergreen. Beautiful tree, grows in that shape and everything. Gorgeous, puts out a cone. But every fall, they turn this beautiful color of rusty orange, and then the leaves all fall off. And then the next year they put out new leaves. Well, when you don't know this and you look and see that the tree has lost all its leaves and you think it's an evergreen and you say, uh-oh, our trees died, we better cut them down. Yeah, that happened to a certain jurisdiction's municipality I won't talk about, cut down 3 of those because they thought they were dead. So it's very important to know what you're dealing with, right? Tree ID is extremely important to an arborist. Anyway, I digress. The needle-like on the right with the conifers, that's a pine, right? That's going to be in the Pinus family. Remember, 2, 3, 5, all kind of stuck together in a little bundle at the base. Okay, the leaf apex, that's the top of the leaf, right? Or the end of the leaf, I guess, if you're looking at it from where it hangs on the tree, there's the petiole, and at the very bottom is the leaf apex, right? So it can be acute or obtuse or truncated or cuspidated. That's always fun. Cuspidated, kind of think of a bicuspid, right? That shape of your tooth. And that'll kind of tell you what that one looks like. But here are just some different examples. And again, this is all going to help you towards tree ID. Truncated, right away, I think, well, that's maybe a tulip poplar has that kind of shape at the very tip of its leaf. But there's different ones, you know, bristle tip. There's going to be, you know, pointy, think of pointy leaves. It's going to have that bristle tip. Now, as far as the morphology of the leaf, compound leaves are going to have those leaflets, right? It's not going to be just one with a single petiole. It's going to have the petiole at the very bottom, like on this leaflet. It starts off with that bipinnate leaf on the left. You've got your petiole, and then as it comes up the tree, you end up with a rachis, right? So it goes from like a bracket in between each one of those little leaflets, right? And you have a pinnule that comes off of the rachis. So off that, you have more little leaflets that come off on a bipinnate leaf, okay? On the palmately compound leaf, palmately, there's a way to remember that because it looks kind of like your palm. That's right. All right, palmately compound leaf. So you've got leaflets that all come off. Look at the picture there, palmately compound leaf. You've got the petiole at the bottom, and then you have all these little leaflets that come off in the shape of a palm, right? All right, and then you have even pinnate at the very bottom of the compounds. You have your petiole at the base, and then you have a leaflet or pinna that comes off in little pieces, little opposite of each other on the rachis, right? You've got the rachis between each leaflet. So the petiole turns into a rachis, and then the little leaflets come off in opposite directions, right across from each other. And that's an even pinnate, okay? All right, so hopefully everybody's following along with that. All right, so let's look at some of these compound leaves. Honey locust on the left, gorgeous color. Has really sharp thorns though, if it's not the thornless variety, but that's a compound leaf. You see all the little leaflets coming off of the rachis, right? At the very bottom, you have your hickory that's coming off in opposite sides. You can clearly see the petiole at the base, and then as it comes to the rachis in between each set of leaves that come off. And then the buckeye, that's one of our mad horse trees. Woo, the buckeye, how it comes off of the rachis and the petiole, and then it puts out each little compound leaf to the side. And they're not always small. You look at that buckeye, that's a pretty long, big leaf. Same thing with the hickory. It's not small little leaflets, it's large leaflets, but they're all compound leaves. All right, so here's a quickie way to remember simple and compound. This might look like something in your book, so you might want to look for that. So there's your simple leaf on the left, right? So you've got a stem, and off that, you've got the bud that comes out at the very base. That's what you're going to look for in spring or in winter. You're going to see a bud scar, and then each spring, you're going to have a new bud. Right? And then you have your petiole that comes off the stem, and then it goes up to the midrib, and that's a single leaf. You have one leaf. It's got a blade. It's got all the things we talked about, the apex and all that. That's your simple leaf. On the right, you have your compound, and it is going to have the same stem. Then it's going to have its bud or bud scar, depending on the time of year. And then your petiole coming off of the stem. And then once you hit leaves, you're dealing with the petiole. Right? The petiole goes between the leaflets, and the leaflets come off as a compound, right? So they're opposite, right? Even, okay? And that all is going to help you identify your tree. All right, so if we're looking at the tree on the right, the image on the right, what do we think that might be? And that is one tree. Notice it's got a lot of different leaf shapes. If anybody said sassafras, you're correct. That's a sassafras leaf on the right. And on the left, we have, anybody want to take a guess? Well, if you do, you can throw it in the chat. If not, that's okay. That is in the holly family. And you can see the little spines that come off of that, right? So leaves look different ways. And I'll tell you an interesting thing about the sassafras. You notice it's got three distinct leaf shapes. You have the big, wide, like flat, normal looking leaf. Then you have one that's split into like a leaf plus a little extension piece, almost like a thumb. And then you kind of have one that goes in three, you know, like the live long and prosper. Anyway, there's a Native American story, which is really cool, that the reason that tree has three leaves is because, you know, first man and first woman, when they were learning how to make fire and make a living, and how to survive in the wild, first man came back and said, boy, I can't pull my bow. My hands are frozen. And first woman said to the creator, hey, in prayer, let me help my husband out. And the creator spoke to her and said, go to the tree in the front. And so she went out and she found this beautiful leaf. And she thought, it's big enough, I could make a mitten out of that. So she sewed two together and gave it to first man. And he went out and took the mittens and said, oh, my hands are now warm, you know, but I can't really pull the bow with this. This is terrible. So he couldn't hunt, came back, and she again prayed to the creator. And again, the creator said, go to the tree. And this time the mitten appeared, right? Had the thumb. So she took that, and now she had a pair of mittens for him with thumbs. So he said, oh, this is great. I'll be able to pull the bow. So he went hunting the next day, and yeah, he could pull the bow, but he had a really tough time trying to knock that arrow. And he went back and said, I still really couldn't hunt, and my hands are still frozen. And so she prayed again and went back out to the tree, and this time the third one appeared. So now after she made those new mittens, he could actually pull the bow, knock the arrow, and was able to hunt. At least that's the Cherokee. I think it was Cherokee legend on that. Anyway, I always thought that was interesting, and it always helped me remember sassafras has three different leaf types, right? So listen to your lore. Sometimes that can really help you ID a tree too. All right, here's a good example. If you said this one looks kind of what shaped? Yeah, it might be palmate, but it's not a palm. So, I wonder what that is. I can see my margins. I can see my apex. I can see a little bit of the bark on the tree. I can see the petiole. That might be a saucer magnolia or an umbrella magnolia. Yep. Things like that help you ID a tree, right? And if you said that it was a umbrella magnolia, you're probably right. All right, a deciduous tree in winter, you're going to need to be familiar with what? Well, you're not going to have leaves. So unless you can find them on the ground, but hey, they blow around. That could be from other trees too. So it's not always going to help you with the leaves, but the bark is always going to be there. The branching habit, how it grows branches, is going to be there. Your twigs are going to be there. The buds or bud scars are going to be there on that stem. The fruit may be there, right? You may still have some fruit hanging around. And the pitch inside that twig is going to be there. So those things help you ID a deciduous tree in the winter. And some deciduous trees, I can think of one off the top of my head, the beech tree is not going to lose its leaves in winter because that bark is so thin, it's going to keep its leaves to keep it from getting sun scald. So there's an ID that might help you out too in winter. Does it keep its leaves? And with them, they push out the old leaf when they put out the new leaf in spring. So that all helps tree ID in winter. But most of the time, you're not going to have a leaf there. So you're going to need to know what that bark looks like, you know, and what the branching habit looks like. So winter bud morphology. Remember we were talking about the buds and where they are? Okay, so on the left is an example of a green ash twig, right? A little stem that has a bud at the very top. It always looks like a little Russian hat to me, or a little hat of some sort. So that makes me think green ash. Like I said, it's all about trying to help yourself remember things so that you can ID these trees properly. If you look over on the right, you'll see the outline of this twig. And you have, when you cut it open, you'll have the pith in the center, and each pith pattern looks different, right? You also on the twig will have a bundle trace or a leaf scar where that bud fell off last time, right? So there's always going to be that little scar there, and you can kind of see that on the midsection of this little green ash twig. You can see a tiny little bud above it, and you can see the leaf scar underneath. That's where the petiole broke off, right? When the leaf dropped. So that scar is always going to be the same on a certain type of tree. It always looks the same on the bud scar. And then there's different things like the ring of bud scale scars from previous year's growth. So where this says ring of bud scale, that's where this top terminal bud used to be last year before it grew out, right? So each year they grow, right? The leaf keeps, the twig keeps stretching out, the tree keeps growing. And each year that terminal bud moves to the end of the twig. There are also lateral buds. You can see they're much smaller than the terminal bud. There are bud scales, those little pieces that hold the bud. So it's really kind of interesting in bud morphology, can really help ID a tree. So that's how that goes. All right, palms, like I said, are giant grass. Woohoo! Thousands of species of palms, more than 200 genera. I still don't remember them all, and I've been doing this for 20 plus years. They're native to all continents except Antarctica. They don't like the cold. Sorry, at least not that kind of cold. Yeah, so they're everywhere except Antarctica. Most are single stem, but some are multi-stemmed. It just depends on which palm you're looking at. Some are shrubby and others are kind of vine-like and grow up. Some can grow up to more than 200 feet tall. That is a really tall palm tree. And most palm leaves or fronds are compound. They're either palmate, remember we talked about what a palmate looks like, or pinnate. And pinnate to me always kind of looks like a feather. Think of a pin feather, and then you can recognize that pinnate leaf a little bit easier, right? All right, so tree identification keys are forms that have been step-by-step methods for identifying plants. It's a little written kind of key, like a book, that describes the shape, texture, arrangement of leaves, the bud characteristics, the twig shape, the morphology of the flowers and fruits. And it kind of has a flow chart with questions that you either say, does it have a palmate leaf? Yes. And then you kind of go to the next thing, right? It'll tell you to go to the next page or go to this page if it's palmate. If it's not palmate, it's pinnate, it'll tell you to go to this page, right? So you're answering these yes/no questions to try and determine what type tree this is. And that was long before we had computers. We didn't have these. We didn't have computers. We had a tree identification key that kind of helped us. So we all had these books that we carried around trying to figure out what was what. Nowadays, you have a computer program that can help you ID it. But this is basically what a tree identification key looks like. So it tells you, if you answer this plant has this, you go to key 2 or 3 or 4, and you keep going down as you're answering them. Oh, well, then now we got to go to page 115 if it has this. So that's what a key is, and they're written. Now, they need a certain level of expertise and understanding of those terms and of the tree parts to be able to ID the plants correctly. All those things we just talked about, you have to know before you can go to a tree identification key to even start trying to get an answer, right? Plus, the plant may not match its written characteristics exactly because of either seasonal differences or plant color or morphology, right? Or the time of year, right? Yeah, so... And they're not all keys have trees that might be found in a given region. Like there might be a tree identification key for here in the Southern Piedmont, but that might not work for out west, right? Because they got totally different trees than we do. So you have a whole different tree key out there than you would here or up north, same thing. You're going to have a different key for every region you're in. And genetic variability, plant conditions, locations, whether it's getting enough water. Is it growing under a bunch of other trees and the leaves are twice as big? Is it still kind of young and the leaves are big? All this stuff in the environment can also affect the size of the plant and the size of the parts and how it looks. So tree identification keys are very helpful, but it's got to be specific to the region you're in, and you really got to know your trees and your tree parts to be able to use them correctly. And I think that's it for me. And don't forget, there are some really good programs on computer apps that ID trees. PlantNet is one of them. Oh gosh, there's Leaf Snap. There's all kinds of them nowadays that really help in IDing trees. But don't forget, you can always go old school. If your computer dies, you might want to have a key of your region so you can help.
Thanks, everybody. Hope you had a good break. We're going to hand it over to Rob. On to you, Rob.
Thanks a lot. All right, so hopefully everybody can see my screen and it's the correct one this time. One of the things that I want to start off saying is that please keep in mind that the material that's presented here is for an international audience. So there are things that you might read or experience in the text or in this presentation that would apply globally. And we might discuss things that might occur locally. Please just keep that in mind that this is an international exam, so it's going to be a little more generic than a localized presentation might be. And just so you know, in the text itself, it is really written with Northern Hemisphere hardwood forests in mind. That might help you out a bit. If you have questions, not only can you leave them here during a live presentation, but if at some point a question pops up before you take your exam, feel free to send me an email. Just to try to put something in the subject line like ISA certification exam review so that it doesn't go to my spam filter. This is the certification guide that you should be working with. It's the green one. There is a new program that's out right now, but it has just been released. But your test will not be geared towards the new exam guide. The content's really not all that much different. It just has some more in-depth figures and things like that. I also highly recommend that you purchase this complete set of the Ansi A300 specifications. You'll notice that there's these parts fairly close aligned with the ISA. The background or the primary source for a lot of the specifications that we'll read about and talk about in the ISA lectures have been derived from these ANSI standards of the A300. So if you want a little more in-depth understanding of a particular subject matter, this is a great place to get that information. And if you look across the bottom of the screen here, you'll see that this is where you can purchase it. You can also purchase it on the ISA website. All right, so introduction to tree biology. So tree biology is where it all starts, and understanding trees, how they work, how they behave, why they do the things that they do, gives us great insight into how to treat them and things like that. So today we're going to look at cell structure and compositions. We'll start at the microscopic level. We'll talk about meristems, the differentiation that gives rise to buds, leaves, wood, and roots. We'll talk about the vascular systems and their different arrangements and functions. Talk about mycorrhizae and the development of the root system of trees. Photosynthesis and respiration, tree growth and development. We'll talk about compartmentalization of decay in trees, a very, very important topic that also is highly associated with the risk management portion of the exam. And then lastly, we'll talk about palm anatomy and physiology. What makes a tree a tree? Well, trees are large, woody plants typically with a single main trunk. They're long-lived, so they live more than just one season. They keep going. They're compartmentalizing, and they keep coming back. They're perennial, so it's large, woody, single main trunk, long-lived, and compartmentalized. Why do we need to know how a tree grows? Well, one must know how things should be in order to know when they're broken. You need to be able to keep that in mind. That am I looking at a tree that has a problem? I mean, what should it look like to begin with? That's what you need to focus on here. It helps to diagnose problems and prescribe the treatments. So if you know what it should look like, and it's a different color or it's got a different shape or something to it, you would be able to identify it because you know how it should look. Understanding that genetics are influenced by external stimulus helps to assess their potential in a given growing environment. A bonsai might be a good example if it's being bonsaied, if it has a restricted root zone that's being pruned, a canopy that's constantly being pruned, yet it might have the genetic potential to be a 50-foot tall tree, but because of the way it's treated, it's not reaching that type of genetic potential. So we're going to talk about anatomy, morphology, and processes. Component parts of a tree. Anatomy. So anatomy is the science of the structure of animals and plants. Morphology is the study of the forms that those things take on. Physiology has to do with the function of these parts. What are the biological processes, chemical and physical processes involved, and what do they do? We're going to start with cells. Cells are the very basic building blocks of our plants, of any living animal, a plant or animal. New cells arise from the division of the existing cells. So a single cell is going to go through a process that allows it to separate into two cells. In trees, division only occurs in very specialized zones called meristems. We'll learn where these meristems can be found. After division, cells undergo differentiation. So when a cell is first divided, this new cell can really be a multitude of things, but the tree gives it the information that's needed for it to become something with a particular structure. So that's undergoes a change there. Cells with similar structure and function are arranged into tissues. So if I'm big into producing xylem tissue where I expand, well, I'm just going to keep producing xylem tissue. If I'm a meristem associated with twigs, well, I'm going to create a shoot elongation and things like that. So they're more likely to produce whatever they're adjacent to. So here's kind of the waterfall look at it. Cells are going to congregate into tissues, and tissues can gather into organs. So like some of the organs might be leaves, stems, roots, flowers, and fruit. Now, if you add all these things together, it gives you a fully functional tree. Over here, we have some examples of where you might find the different organs. So I've learned that on the exam, you're not likely to see figures. So it helps to set these things in your mind. If you can picture and carry these pictures with you into the exam, it might help you out some. So let's talk about those meristems. Where are those cells dividing and reproducing? So there's two types. There's going to be primary growth and secondary growth. Primary growth produces the cells that result in elongation of shoots and roots. Growth of shoots and roots occur at what's known as the apical meristem. Apical just means the apex or the very tops, or the apical or the top or the end. We have apical meristems. That's how things get longer. The second kind of meristem is secondary or lateral growth. This is what, when I was a teenager, I was 112 pounds, but I was about 6 feet tall, and now I'm still about the same height, but I've gotten a lot bigger than 112 pounds. So I grew in secondary or diameter. Meristems within the stems, branches, and roots all have secondary. So once elongation is complete, everything's going to get a little bigger. So I like to think about it as a new tree grows on the outside of the previous tree every year. So it's adding vascular system. It's going to add some bark. It's going to add diameter every year. It's what produces the wood. It's what allows trees to grow large. And the thing to point out here is that palms lack secondary growth. A palm is going to be the same diameter when it decides to begin shooting up as it will be when it's 30 years old. It'll still be that same relative diameter. They do not have secondary growth. They just have shoot growth, so they just get taller. Here's a close-up look. This is a plant root. This is this little point where you see where each one of these little blocks that you see around the edge. These are cells. You can see where the cell size gets smaller and smaller as you approach the end of this red line. And that area is the primary meristem. That's what's going to produce the material for this root to be able to push through the soil. On the outside of this meristem, we have what's called the root cap. The root cap is kind of hardened. These cells out here can actually produce a lubricant that is exuded into the soil to help it push through the soil as these new parts are produced. So apical meristems are very small compared to the cylinder-shaped lateral meristems. Again, these primaries are going to be located at the very end of a branch or at the end of a root. Here we have an example of the lateral meristem. Here's the cambium that we've probably heard about before, especially if you've studied your book before taking the class. So this cambium is the primary producer of lateral development. So it's going to produce xylem tissue to the inside, phloem tissue out towards the bark. Here's some more about lateral meristems. We have the cambium here. Again, this highlighted yellow area is the cambium. It's going to produce xylem tissue to the inside. And we'll learn more about the function of these shortly. Phloem tissue to the outside. Now, once you get past this phloem tissue, well, then we get into bark. And so here's some bark. We're going to have a little bit of cortex here, cork cambium that's going to produce phylloderm to one side. And then all of these cells on the outside, these are all dead. So just keep that in mind. Again, just sort of think about the sequence that things occur from the center of the tree going outwards. And that'll help you during your exam to be able to picture where these things are. Cell walls in plants are significantly different than they are in animals. Plant cell walls are hard. There is no cellular replacement in a plant as there is in a person. Like if I get an injury on my skin, if I cut myself, my body's immediately going to start sealing up the wound, and it's going to begin a healing process where the material is going to be replaced. It's going to take out the damaged material and it's going to start replacing it with good healthy tissue. This does not happen in the plant world. Once a cell is dead, it's always dead. It's not replaced. And this is why we need to take the word healing out of our vocabulary when it comes to plants. Plants are not capable of healing. So please try not to use those words. Cells are made up of both cellulose and lignin, and this is what gives them that strength on the outside cell wall. So cellulose is a structural component. It's the most common organic compound on Earth. It provides the architecture of different types of cells. Lignin is what gives rise to strength and rigidity. Lignin is what allows trees to get to be tall. Let's take a look at xylem. So the primary function of xylem is that it conducts water and dissolved minerals from the soil up into the canopy. It also supports the weight of the tree. So it bears the compressive weight of the tree. The most common stressor a tree experiences is gravity. The next is probably wind. So it has to be able to support the weight of the tree without crushing itself to death. Xylem is also where a good number of carbohydrate reserves are stored. The ray cells can get out there and store carbohydrates or retrieve them. We'll see how that happens shortly. It also defends against the spread of disease and decay. The xylem tissue, the living xylem tissue, is responsive to injuries. So it becomes a critical part of the coded model that we'll talk about. Complex tissue, so xylem, there's only so many growth increments which it remains alive and in a capacity where it can store those excess starches. On the inside of that, I'm sure if you've cut a tree down, a large one, and look down and top, you see this bright white wood, and then you see a darker wood. And that darker wood is old xylem tissue that is no longer living. And so when we talk about xylem, you might want to discuss it in terms of, is it living tissue or is it non-living tissue? So symplasm or apoplasm in the tree. On a large tree, a big portion of the interior is going to be this non-living apoplastic tissue. And so xylem can take on slightly different roles and appearances depending on if we're looking at an angiosperm or a gymnosperm. Let's take a look at the gymnosperm first. Tracheids conduct water and provide mechanical support. They are elongated dead cells with pointy ends and thickened walls. Again, these are parts of or contained in the xylem tissue. Fibers provide mechanical strength. There are fibers that are interspersed inside of the xylem tissue. That's what gives us some of that compressive capability. Parenchyma, these are living cells that are interspersed among other xylem cells. They store carbohydrates in the outer layers of the xylem, again, the live part. They can help defend against decay, and they have a structural function across the grain. Here we have a cross section of a piece of pine showing the parts. There's 3 annual growth increments here. This line is a demarcation between fall and spring. And again, we have that same line of demarcation here. So from this point to this point is one complete growth increment. Notice there are some cells here that manage to cross over that boundary layer. Again, those are going to be ray cells made up of parenchyma things. Here's what it looks like on an angiosperm or a hardwood. We have kids, but they're not quite the same. We have fibers that hold everything together. Parenchyma is a lot more abundant in hardwoods and arranged close to the vessel elements. Vessels are the primary conducting elements. They are stacks of dead hollow cells that form long tubes of water conducting elements. These are more efficient than tracheids. So we have some capacity with tracheids, but a lot more in these open vascular vessels. Now, the arrangement of these tissues inside can occur in two different ways. It can occur in ring porous arrangement or a diffuse porous arrangement. Ring porous has fairly wide vessels that are formed early in the growing season. Think about, okay, there's lots of rain available. I'm going to do my best to utilize the water that's in the soil and get as much out of it, and pump it up into my canopy as possible. However, later in the season, the water might not be so available, so I'm going to make smaller and smaller vessels so that I don't waste as much energy trying to get water out of the soil up into the canopy by producing large vessels. What types of trees are ring porous? Well, oaks are going to be the most common, elm, ash. Diffuse porous, these things form uniform sized vessels throughout the growing season. So there's not really a big difference between the beginning of the growing season and the end of the growing season within the growth increment. And why do you think that might be? Well, maybe they have rain or soil moisture available the majority of the growing season, such as a swamp. So notice that these trees that do well in swamps, maples, plane trees, and poplars, are diffuse porous trees. That doesn't always hold true, but that is primarily what happens. So here's an example of a ring porous tree under some microscopic examination. So this would be the spring. Notice how large these vessels are in the springtime. And then as it grows throughout the season and adds on more vascular capability, note that it gets smaller and smaller and almost disappears by the end of the growing season in the fall. Compare that to a diffuse porous tree where the size of these vessels are about the same diameter beginning to end. The staining process here closed up some of these vessels, makes them a little harder to see, but they're basically the same size. So here they're obviously large, and these tiny little ones here are small. So if you look at the size of this one and compare it to the size of this one, they're going to be about the same. Here's another example, a visual example for you to show the difference between heartwood and sapwood. Again, this portion of it is going to be dead xylem cells, and this portion out here will be living xylem cells. So we also need to consider what is a growth ring. Again, this is the increment or the amount that a tree grows from season to season, provided the tree actually has seasons. It is found only in the xylem tissue. So think about for a palm for a few minutes, do palms have secondary growth? No, so you can't cut a palm down and look inside of it and see these annual growth increments. So this is something that you're only going to find in the temperate forest regions of the world. They are visible in a cross section. Again, so if the tree's growing up like this, the cross section is going to be this way, not this way, but this way. And it's the result of the seasonal production of xylem by the cambium. We have what's called early wood, and again, those are the cells that produce early in the season. So this would be an example of early wood, these large vessels, and then late wood, the vessels are going to get a little smaller and more compact. They will get a little more compact here at the end of the growing season in the diffuse porous as well. It's just a little harder to see, but there are early wood and late wood cells that are produced. Conifers do not have vessels and therefore are non-porous. Growth rings may not be annual for tropical trees. Again, remember I said it was based on seasonality. So the closer you get to the equator, the less seasonality appears and the less likely you are to see growth increments in a cross section. There are still constantly produced vascular tissues that show up on the xylem side in tropical trees, but you just don't see this this start, stop, start, stop, start, stop like we do here in the diagram. So not all conducting elements transport water. So just because all of this is sapwood does not mean that it absorbs water out of the soil, moves it up into the canopy to be used in the leaves. That doesn't happen all the way through here. It really just happens in the outer ring or so. Conifers are able to do it. Remember, they don't have vessels, they have all tracheids. 2 to 12 rings may conduct water in a conifer. And in hardwoods, usually only the outermost 1 or 2 rings conduct water. You say, well, why 1 or 2? If at some point the tree stops development of new vascular tissue, but it doesn't kill that tissue, it just stops growing because it's winter, and then it has to pick up again in the spring. And so in the springtime, it begins manufacturing more xylem tissue as the weather warms up. So for a little while, it is going to use both rings. There'll be both rings will be conducting at the same time, and then at some point it shuts down the second inner ring and relies strictly on just the first ring. There are a few trees out there, however, that can maintain 2 growth increments of water conduction, such as an elm. Sapwood that conducts water contains many living parenchyma cells. Heartwood, non-conducting to tissue, contains no living cells and can sometimes be darker. So this dark color that you see doesn't always have to exist for it to be heartwood. It's just commonly thought of. So let's switch now from the xylem tissue, which is produced to the inside of the trunk, to phloem, which is produced towards the bark side of the cambium layers. So what does phloem do? Phloem moves the carbohydrates produced in the leaves throughout the plant for storage or consumption. So phloem is going to take everything that's produced out here in the canopy, and it's going to move it inward and downward until that energy is consumed or excess is stored. Movement is relatively slow and can occur across pressure gradients. It requires energy, so it's an active process to be able to move these things. You might think, wow, you think gravity would just suck it down, but it doesn't really work that way. It's composed primarily of living cells. Remember, the xylem tissue, the actual vessel itself, is typically not alive. So in conifers, for the phloem, we have what are called sieve cells in conifers, and sieve tube elements and companion cells in hardwoods. So here's an example of one of these sieve cells in a hardwood. It has very tiny holes in the top and bottom, so it makes it look like a sieve or a colander. So think about it as a sieve cell. Notice these arrows go in both directions. So it's important to understand, especially when we get to the pruning section, that the majority of energy moves downward. It moves down the leaf, down the stem, down the leader, down the trunk, and out the root. So it's moving like this. Very small amounts of energy may be able to move up, but that's not going to move from, say, the trunk into the upper canopy. It's more likely that it's going to move from a little leaf here out to feed an apical meristem or a bud for next year, or it might be very localized movement that it moves upstream, but the wholesale movement is definitely downstream. So here's a look at a xylem vessel again, and compare it to a phloem vessel. So that these are dead cells that are stacked on top of each other, and they're totally open top and bottom, so it's like a giant straw. This one can move not nearly as quick. It's because of the sieve cells are going to help slow it down and help the tree to manage These companion cells can say, hey, I need some energy out here in some adjacent tissue, so it can notify this cell and can move across the membrane and move storage or move excess photosynthetes out into a storage area or an area where it's needed. Old phloem or non-functioning phloem gets crushed out here into the cortex and can be reabsorbed into the cell walls and incorporated into the bark. So there's not, like with xylem, there's an annual accumulation, and so you get those growth increments. With the phloem, it's constantly producing phloem tissue, and as it ages out, it's crushed into the cortex where it's reabsorbed and used in the phylloderm, which is going to produce more stuff out here. Let's talk a little about the transport system. So we talk about axial or longitudinal in the phloem and the xylem. We have radial. This extends across the growth increments. Rays are composed of parenchyma cells arranged in small layers. So again, we have phloem tissue primarily is going to go up and down. It's going to go up or down. The ray cells are going to move things across the grain. So we have axial or longitudinal, it's with the grain. Radial will be across the grain. Ray cells fall into that category of going across the grain. What do ray cells do? This is one of the very important features, again, in the coded model. We'll talk about this again. But for transport systems, let's think about rays as being able to transport carbohydrates and other compounds into the sapwood. Remember, sapwood is that living portion of the xylem tissue. So if I'm constantly producing energy out in the leaf tissue and I'm trying to move it inward and downward, probably have cells that are right around where I'm being, right around the leaf, that are going to consume some energy, and then maybe the twig needs some to stay alive, and then some's going to go down the branch and get stored there as it goes. Eventually, it's going to get either used or stored somewhere along the line. Trees produce about 66% more energy than they consume. So consumption really only, on a healthy tree, only represents about 33% of the living part of the tree. Carbohydrates are stored as starches. It's a fairly stable compound. Sugar is a fairly stable compound. It doesn't want to degrade into something else readily. So the trees store it as this carbohydrate. Ray cells also assist in restricting decay in wood tissue. So this becomes sort of this ray cell can become a boundary. This one can become a boundary. We'll learn how that works a little later. But these are living channels that can move things in and out. It might say, well, we need to store some water, so it can move some water into here. It might say, well, we have open storage space here for extra photosynthate. Let's move it in towards the center of the sapwood, and store it. So let's move out to bark. So bark is the outer covering of the trees. Upper portion, and can sometimes be found on the root system as well. It's composed of protective, non-functional phloem and corky tissues. The amount of corky material varies by species. If you think about American beech, has a very, very thin layer of bark, so would a maple or a magnolia, things like that, have fairly thin bark. But take a look at the Spanish cork tree, and it might be several inches thick. Think about in a wine bottle, those corks come from this area. That's where it's produced, this sort of corky part. So you can see that the thickness of that corky material can vary greatly. The cell walls of the bark are impregnated with waxes or oils. This helps to minimize water loss and insect damage. It can also moderate the temperature inside of the stem and provide obviously defense against mechanical injury. The chemistry that goes on in photosynthesis is remarkable. It requires the combination of different chemical compounds and elements to produce this photosynthate. And all of this chemistry produces byproducts. We all know by now that the process of trees, photosynthesis can give off oxygen that we breathe. The root systems can give off carbon dioxide. So these are residual gases that are left over after the combination or separation of carbohydrate molecules. We can't just have an abundance amount of gas inside of the wood tissue, or else the wood tissue would start to break apart. So it has to have a way to vent those gases. It also might need a way to absorb those gases. And it does that through what we call lenticels. They're small openings in the bark. They're found on the outer surface of the stem and twigs. They permit gas exchange with the atmosphere. And sometimes they can look like quirky protrusions on the stem bark that allow for gas exchange. In the example shown here, you can see these lenticels, these little white dots, are very small. If you stop and think about a cherry tree, like a Yoshino cherry, you can probably see those big splotches on the leaders and on the stems from 15 or 20 feet away. Those also are lenticels. They are tremendous in size, and so the size of these lenticels obviously is quite variable. Stems are the small portions of the branch that provide support for the leaves, the flowers, and the fruit. Again, we're going to step down. So the branches support the twigs, and the trunk supports the branches and the twigs. So just think about that hierarchy. Kay showed us this diagram earlier. She was speaking from it as a way to key into the identity of a tree. Now we're going to see some other things that this stem anatomy is responsible for. So now that we know a little tree biology, we can apply it. So here we have what we know as the terminal bud, or the apical bud. So you could say terminal or apical, it's the same thing here. It's normally the most active on each branch or twig compared to the rest. We also have lateral or auxiliary buds. Here's an example of lateral buds coming off. So these would produce either a leaf coming out, could produce a little stem coming off, something like that. But it's not going to grow as much as the terminal bud, especially during shoot elongation. Lateral buds are often dormant, growth inhibited by the apical dominance of the terminal bud. So believe it or not, this little bud out here produces a chemical compound that retards the growth or development of these lateral buds. So in the absence of that limiting compound, these other shoots can grow quite vigorously. And we'll talk about that effect when we get to pruning. As shoots lengthen or age, or when pruning removes the terminal buds, dormant buds may become active and form shoots. And that's what I was talking about. So in the absence of that hormone, these things can shoot off. So a node, this is a slightly enlarged portion of the tree. So you can see where this scar looks really different from anything else on this stem. This scar that goes all the way around is referred to as a node. These nodes start, so like the following year's node would be right here. And so this part will go through stem elongation and grow some distance to the end of the next growing system, and there'll be another node up here. So the distance in between nodes, so in between here and up here, is the internodal area. So this is all internodal. This in here would be internodal. So when they talk about node length, they're talking, or the internodal length, they're talking about the distance that it grows from here to here. If you have a long branch and you can examine that branch along several nodes, you might see that the internodal length is a few inches every single year. So that means the tree's in a constant state of growth. And if they're getting a little longer every year, it means that perhaps you started fertilizing the tree, or water was more available that year, and so it was more conducive to growth, so it grew more. However, if you go into a drought or you stop fertilizing or something like that, you might notice that the internodal distance gets shorter year after year after year. So that's something to look for as a diagnostic for health. Here we have adventitious buds. These buds, like we said, they're often suppressed and they do absolutely nothing. We have secondary growth every year. Remember we talked about secondary growth, or the getting larger in diameter. So over time, these buds sort of get absorbed, but they hang out just underneath the bark is what we know as a meristematic point, or a latent bud. So these latent buds, meaning they're going to lay around and do nothing until called upon, or something's going to trigger those at some point. It could be over pruning. It could be some type of stimuli is going to cause this adventitious growth development. So there's no telling what caused it here. The tree could have been topped. It could have started to fall over. It could have had freeze damage in the vascular tissue up here. And so now there's no hormone that signals those things to remain latent. And so they explode and can start to grow. Another type of terminology that we use here is epicormic. Cormic shoots result when dormant buds elongate. So dormant buds, again, latent buds. So these things, we call it adventitious growth development, adventitious shoots. The more technical term is epicormic shoot, but you could see all of these. Again, on the test, it's not likely that you'll see things that are all of the above kind of answers. It's multiple choice, but you might not have heard from several people now that they don't have all of the above answers. So there might be a couple of questions that are pretty close. So if you can apply what you have learned here to the questions, so read the questions very carefully, then read the answers very carefully, and look for the tricky stuff. So there might be an instance where you'd see epicormic growth be the correct answers. Branches, when they grow, so we're going to talk a little about morphology now. Remember, morphology is the appearance that things take. Branches are autonomous. They produce and store enough carbohydrates to sustain themselves and export some to the trunk and roots. Remember when I said that the photosynthates didn't move up very much? Photosynthetes are going to be contained within a twig, within a branch. And so they're really not going to be able to move them out from the base of the branch out towards the tip of the branch, or from the base of the branch up to the next branch up or into the upper canopy. So it's important to know that branches have to operate autonomously. So you can't prune so much off of a branch that it loses all of its energy, because then the branch is dying because you can't produce enough photosynthate to feed this big sink. We'll talk about sinks and sources later. They do not import carbohydrates from other parts of the tree. They have a unique attachment that we'll look at under great detail when we talk about pruning. Branches are strongly attached to wood and bark below the branch, but kind of more weakly attached above the branch. And so on the top side, so wood is much, much stronger in tension strength and weaker in comparison to compression strength, which would be weaker. So you have to overdevelop compression wood and compared to the tension wood in order to hold something onto the tree. Layers of tissue produced annually at the junction. Remember, again, we have this sort of lateral development every year. It's going to get bigger. So we're going to have these layers of tissue that are going to overlap a little bit or bunch up some. It might create a branch collar. At least we hope it will produce a branch collar, which produces that sort of bulge or shoulder. As you have the stem tissue overlapping with the trunk tissue, you're going to have that little bulge area where the two tissues overlap each other. Where the branch and trunk expand against each other, the bark is pushed up, creating a branch bark ridge. When the bark is in a crotch surrounded by wood, it can become included bark. This picture is an example of included bark. Take a look at the branch closer now. So here you can see this kind of shoulder, this very clear area right through here. This sort of fattened shoulder, that is the branch collar. And this is the branch bark ridge. So if we follow this branch bark ridge all the way down to the bottom, this is basically the diameter the tree was when this branch started growing. And I know this can be a little difficult to think about, so I'm going to try to paint some stuff on here for you to see. So if we come in, maybe it's not going to work. Sorry about that. I don't think it's going to work for me. So think about it, the lateral development here. If this branch is an inch thick, it might be, say, 3 years old. So if we look back in time and think back 3 years, it's going to look more like this little twig that's sticking out on the backside. This is its first year. It's going to be that diameter. Well, during that same 3 years, this trunk grew in diameter. So maybe 3 years ago, the trunk was only this big in diameter. And all of this is new tissue that's grown about since. So if we think about that stem tissue growing in diameter and the trunk tissue growing in diameter, that's where it forms this branch bark ridge. That's how it's formed. If we look over here, we give a sort of a preview into pruning, if you will. So this would be the proper area to cut because we want to minimize our decay. If we made this cut at the branch collar, we're going to only damage limb tissue. We're not going to damage any of the trunk tissue. So you never want to cut this off because then you're getting branch tissue and trunk tissue in the same cut, making decay, massive decay much more likely. Now that we've got branches and twigs and all that sort of stuff, let's look at the leaves. In a deciduous tree, leaves are disposable. So we don't have to spend as much time with these elaborate arrangements of vascular tissue. It can be much more simple. So we create what are known as vascular bundles. So you can see an example of the vascular bundles. This is a combination of xylem tissue along with photosynthate moving materials here. So we can move everything in and out. It's closely associated. We're going to have to shield ourselves from the sun. So we don't want the sun just to dry us out or to burn us up or make us too hot. So there's a thing on the top of the leaf called a cuticle. Cuticles can be very thick, especially in primary canopy trees. Things like oaks, Magnolia grandiflora. Hollies also can grow out in the open, bright sun. They have that very thick cuticle. But then think about a dogwood, something that has a very soft leaf. It's meant to be an understory tree, so it's meant to grow up in the shade. Taking it out in the sunlight, you're going to get some scorch issues or a taco leaf. Again, here's our vascular bundle. And surrounding all of this, we have parenchyma cells. Out here is where the actual photosynthesis is going to occur. And remember, there's a lot of chemistry that goes on inside of this leaf. So again, we're going to need to be able to open and close. So this makes it different from a lenticel. Lenticels are kind of a passive gas exchange. These stomates are active. It takes energy to open and close them. They are surrounded by these guard cells on each side, which can swell up to close the hole or shrink to allow gas exchange. When a tree starts to wilt from the moisture, it can close these stomates to try to minimize water loss. Or if it's actively making photosynthetes and there's lots of chemistry going on, lots of processes going on, it can open that up for gas exchange. The energy of the sunlight is collected in the chloroplasts, which are the green stuff out here. This is where light energy is converted into chemical energy and produce carbohydrates in a reaction called photosynthesis. Leaves are also where transpiration occurs. Transpiration is the loss of water vapor from leaves, which helps to cool the leaf and draws water up through the xylem. Remember, the xylem is like a long straw. If I'm standing a column of water, I need that column of water to be continuous. I need it to have some method to pull it up the straw. Science would suggest, or physics would suggest, that we really wouldn't be able to have a tree taller than about 15 to 30 feet based on atmospheric pressure alone. We obviously have trees much taller than that, so we need a mechanism to draw that water up past the ability to overcome gravity. And so this is the process we think, or the process we identify it as, is called transpiration. So the leaves lose a lot of moisture all day and all night, and that loss produces a pull on the next water molecule behind the one that's being evaporated, and so forth and so on, all down the xylem tissue. And so as that happens, you have this sort of continual upward movement of water. Transpiration again can be shut down and the water can just be stuck in there, but it's closed at the leaf. Leaf blades provide a large surface area for the absorption of sunlight. So you'll notice that their leaves are pretty large, they're flat, so that no cell is very far away from the surface. So they're very efficient at producing carbohydrates. The outer surface of the leaf is covered by a wax layer, again called the cuticle. The cuticle minimizes desiccation of the leaf. Desiccation is water loss or drying out. Small openings in the leaf called the stomata control this loss of water vapor and gas exchange, like we talked. You need to pull carbon dioxide into the leaf for the process of photosynthesis, and water vapor is released because we're trying to get that transpiration to occur. Guard cells regulate the opening and closing of the stomates. Leaves have a network of conducting tissues, including the veins or vascular bundles. These bundles are comprised, again, of both the phloem tissue and the xylem tissue, so we don't have to worry about the xylem tissue in the leaf being stored. So again, it's a disposable part of the tree. We're going to lose it at some point, so there's no need to store anything there. We're going to transport water and essential elements through here and carry carbohydrates that are produced in the leaves to other cells and parts of the tree.
Hey, Rob.
Yes, ma'am.
Sorry, I wanted to interrupt real quick. Daniel Foster has a question. Does transpiration occur during the dormant months as well?
The water becomes fairly static. Things do. This is where that's an excellent question, by the way, and it's one that we still work and sort of query over. But there is transpiration. Transpiration is a necessary part of homeostasis, or the use of energy. So the tree doesn't die. It doesn't become really 100% dormant, so it has to carry on all the processes in order to stay alive. So transpirational pull might not be as significant. It might not be the pressure, the vacuum might not be as expressive when all the leaves are off, but it does still occur. And we're assuming that it can do that maybe through the lenticels or through some other mechanism that we don't fully understand yet. I think you muted yourself again there, Brittany.
Sorry, I don't ever want you echoing on yourself. If that didn't answer your question, yep, he said that did it. Thank you so much, Rob.
Okay, great. So here's a close-up, the underside of a leaf over here. You can see the stomates, the sort of opening in the middle, and then on the outside of the stomate are the guard cells. Again, that's what swells or shrinks to open or close that. Here's the palisade cells, the mesophyll, and again, this is where photosynthesis is going to take place. When photosynthates are produced, they're going to move back into the phloem tissue here. When more moisture is needed or water is needed, it's going to pull more up through the xylem side of this vascular bundle. And again, there's lots of chemistry, very active chemical processes occurring inside of this leaf. So we don't want our leaf to explode because it can't get out all of these gases. So there has to be some little spongy area that's going to absorb for gas exchange, basically. So we're going to have some things that are being produced, some gases being produced and some being consumed. The excesses vent out the stomates, and you can also move carbon dioxide in. So this can't really be solid all the way through, or the whole process would fall apart. So some trees keep their leaves all year round and some do not. As Kay talked about, we can divide them into deciduous trees and evergreen trees. So we can have evergreen oaks and deciduous oaks. These are not necessarily oaks, but you get the picture. So evergreen means that it's going to hold leaves for more than a year. So a southern magnolia growing in the south might have several years of growth on the stem. Or pines can have multiple needles, multiple years worth of needles on the stem. There's some pines that hold, I think, 25 years worth. Foxtail pine, maybe. I can't remember exactly which one holds it the longest. I was thinking it was the foxtail because it gives it this nice bushy look all the way down the stem, making it look like the fox's tail. But pines and magnolias are still going to drop some leaves, but they're going to retain more than a year's worth, so that makes them evergreen. Trees that lose all their leaves every year are referred to as deciduous.
Hey, Rob.
Yes.
I have got one more question for you. Just to elaborate a little bit about the guard cells. What time of day do guard cells and stomata open? Night only, early morning, at what temp? and certain temps vary, pending the variety.
Yeah, the answer to that is really complex. They can open or close at any time of the day or night. The opening and closing can be based on temperature differentials. It can be based on humidity. So there's a number of things, but they can really operate at any time that they're needed to operate in order to keep the leaf cool, to keep it from losing too much water vapor, or when oxygen or carbon dioxide needs to be taken in or vented off. Hopefully that answers the question.
Well, thank you, Rob. Of course, Jason, drop it in the Q&A if you have any other further questions about that.
So let's talk a little about color change. Color change hopefully only refers to the deciduous trees. Trees are going to have to drop this leaf off. If you remember back to Kay talking about leaf scars and bud scars on that stem diagram, that place where it falls off is the abscission zone. And it's sort of a built-in place on the stem so that when the leaf falls off, they can close that down and doesn't lose sap basically through that open wound. So we want it to close that wound off actually before the leaf falls off. The abscission zone is going to protect that area. Leaves are shed in response to day length, temperature, and/or rainfall. So as we get into less and less day length, more and more sun, or less and less sunlight throughout the day, that's sort of going to begin to trigger a response to change that, to start extracting those important compounds out of the leaf and back into the stems where they can be reused the following year or for other chemical processes. Environmental changes cause cell changes and growth regulators to form in that abscission zone at the base of the leaf stalk or at the petiole. Remember, the base of the leaf stalk is called. Fall foliage color changes as a result of the breakdown of the chlorophyll. Remember, chlorophyll is a very complicated, large molecule, and so it begins to break down into its components. Think about, taking down a scaffolding and moving it to the next job site. The boards might come off, the metal parts come off, the foundation comes out. Well, if each one of those were a different color and you added all those colors up, they might be green. And then as you start to pull one of them out, well, it's not that color that was there is no longer there. So then the color of the scaffolding might change. So same thing with the leaves. So the leaves are going to change their color as these compounds are moved out. These reflective colors are moved out and leaves you with only the reflective colors that are still there until the leaves are depleted of those compounds. What's going to make a really good leaf season? It's going to be short, sunny days, cold nights. These things can help with the accumulation of sugars that trigger a decrease in chlorophyll production. So the longer and more drawn out that process is, the brighter the leaf colors are going to be. Here's just a quick example of something, or the compounds moving out of the leaf. It's going to do it in sort of regional areas. So you could even see a leaf with multiple colors on the same leaf as it begins to move those compounds out. Now let's talk about roots. Roots are going to grow where there's two things. Roots are opportunistic. They can only grow where there's air and moisture. And it can't have too much of either. It has to be in the right amounts. There are four primary functions of the root system: anchorage, absorption, storage, and conduction. Here we have main or large woody roots. These are going to anchor, store, and conduct. They bear compressive weight. They might be under large tension. Basically, these are the roots that you can see, whether it's as big around as my pinky or as big around as my torso. These are all main roots. When we talk about absorbing roots, these are going to be very small. This is where the fibrous tissue is found. It can grow along the ends of or along a main woody root, but it's a different classification. They'll be obviously different. Absorbing roots have epidermal cells that may be modified into root hairs. Root hairs aid in the uptake of water and minerals. Remember, the more surface area that you have compared to your overall size, the better you are at absorbing things. Root tips contain a meristematic zone, as we saw in the picture at the beginning when we had it under the microscope. Most absorbing roots are going to be found in the upper 12 inches of the soil. Again, this is a generalized thing. So this is an international exam. This is a perfect example of it. If we went up into the northern hemispheres where we have glacial till soils, they're going to grow a lot deeper than 12 inches. If we have heavy clay soils, well, this might be 2 inches or 4 inches is where these absorbing roots are going to be found. So we like to say that 90% of the root system is found in the top 18 inches of the soil, and 90% of those are in the top 4. So you can see how the majority would be found in the top 12, or a bulk of them would be found in the top 12. We have lateral roots and sinker roots. Horizontal lateral roots are usually near the soil surface. These are going to be these big things, big lateral roots moving away. Some species of trees form these sinker roots. So it goes out some distance and then it'll drop a sink. But it really can only exploit whatever the soil characteristics are. And we'll see that a little bit later. So if our clay soils, if you can't find oxygen below 18 inches deep, say in the Piedmont region of Georgia, then you can take this same tree over here from Australia that does produce sinker roots, and it's not going to produce them up here in our soil environment, because the roots can't live where there's not, remember, air and water. So it has to have air and water to persist. Taproot. This is a downward growing root. It's usually a juvenile feature. It's choked out by other roots over time, that sort of thing. But the taproot has a pretty cool function. We talked about these apical meristems and how they produce a hormone that limits the growth of everything else. So when acorn first begins to crack open and emerge, it's going to put up a shoot for leaves, and it's going to put down another shoot for a root system. That initial shoot, that is the taproot, and it's going to continue to grow downward until it reaches an 8% oxygen gradient in the soil. Well, at 8%, the roots don't live very well. So in Piedmont region of Georgia, again, that 8% gradient is probably going to be about 19 to 20 inches deep. And so that tip is going to die. When that tip dies, that hormone is no longer produced, and the lateral development of the root system can occur after that because that limiting hormone is now gone. Again, taproots are a juvenile feature that fade away over time, again, because the actual root tip or the taproot tip died at 8% oxygen gradient. Very few mature trees actually have taproots, regardless of species. Above the ground, we talk about where you see these flares that come off. This area has all kinds of names to it. Make sure you know what the various names might be. Here we're going to call it the root crown. Roots spread out from the root crown and decrease in diameter quickly in the area of what's known as the zone of rapid taper. So remember that wood is very weak in compression strength. So the heavier the tree becomes, the more compressed this root crown becomes. So the root crown is almost always a little larger in diameter than the trunk of the tree because it's having to spread that compressive force onto the soil. We now refer to this area as the structural root plate, or the structural root plate. And so at the edge of the structural root plate, roots are going to grow from maybe being this big in diameter to being this diameter over about a 12-inch period or so, and that is the zone of rapid taper. So if you've ever seen a tree fall over after a storm and you see that big disc of dirt that hangs out of the ground, that disc of dirt is the structural root plate. Where it broke was the zone of rapid taper, unless there were diseases or something else involved. They become, so on the outside of that zone of rapid taper, meaning distally away from the tree, they're going to be fairly small in diameter, 1 to 2 inches, and they can run the height or 2 or 3 times the height of the tree in all directions, or even further in some situations. And those become what we know known as tension roots. So if we have our tree here and our tree begins to lean to one side, it's going to compress over here on this side of the compression plate, and then it's going to pull on the roots on the backside that are under tension. That combination of forces is what holds the tree up. We'll talk a little more about that in risk management. Roots may extend laterally for long distances depending on the tree and soil conditions. Again, if there's lots of air and water in the soil, roots are going to proliferate. For trees grown in out in the open, roots can extend 2 to 3 times the radius of the crown in all directions. Root extent and direction is a function of the environment rather than genetics. So keep that in mind. If you have a home foundation that was built in the 2000s, the compaction specs under there are going to make sure that there's not about 1 or 2% oxygen available, meaning a root's never going to grow under that foundation. So it's not, if you have a tree planted up against the house, 100% of the critical root zone is going to be found outside of that foundation because it won't be under there. So they grow where it's advantageous for them to grow, where there's air and water. So these fine little root hairs, yeah, they're pretty small. Yeah, they do a pretty good job of absorption, but they're not really good at moving large molecules. Things like phosphates are a pretty big molecule, and that root hair has a hard time sucking it through, crossing it over the membrane. So we have this little symbiotic relationship between a root and a beneficial fungi. This is a symbiosis or symbiotic relationship. The fungi derive some nourishment from the roots. Remember, we have this excess of energy. So after everything's stored, well, it's still going to liberate some to the mycorrhizae to keep them happy. And they are good at things like moving these large molecules. They're also good at absorbing water. They're also very good at growing. If you've ever been out in the woods in the fall and you pull back the leaf litter and you see this little white stringy stuff all through the soil, all through the leaf litter, those are mycelial growth from the mycorrhizae, and they do a great job of harvesting water and transporting other chemical signals. So here's an example of this little thing. Notice this little section almost totally underneath my red dot there. You see this little piece coming off of it. That is a root hair. So that is a specialized component of this epidermal layer of the root. So under about the same amount of magnification, this is an example of a mycorrhizal structure on what's called an ectomycorrhizal structure. Mycorrhizae occurs both endomycorrhizae and ectomycorrhizae. This is an example of an ectomycorrhizal. I doubt you have to know the difference between endo and ecto, but just keep that in mind that they're both types. And so this thing is pretty meaty. That mycelial growth occurs as shoots off of this main structure. So some physiology, photosynthesis, putting together with light is what it means. The process by which green plants use light energy to build carbon molecules that make carbohydrates. Raw materials that are needed are going to be carbon dioxide and water. Carbohydrates is the chemical energy used for growth and development or stored as a starch for later use. When you go to use it, oxygen is released through the stomata as a byproduct of photosynthesis. Now here's a very simplified version of it. Carbon dioxide goes in, sunlight goes in, water goes in. The residual is going to produce oxygen, so the oxygen is going to come out of those stomates, and then photosynthate is produced to the inside. When we talk about energy, energy comes in the form of a sugar or a glucose. Photosynthesis does produce more than just glucose. Photosynthesis, there's tons of chemicals that are found inside of trees. So the photosynthetic process also makes these other compounds as the tree needs them. The building blocks for many, again, these are the building blocks for many other compounds that are required in the plant. They combine with other essential elements such as nitrogen, potassium, sulfur, and iron to create protein, starch, fat, growth regulators, amino acids, and other important compounds. Everybody says, I've never seen a tree with fat before, but I think about an avocado, that fruit out there, the avocado that grows around the seed, that is a highly fat, that is a lipid. So trees can get fat, produce all kinds of compounds. So respiration. Now this is the process by which these carbohydrates that were stored are now going to be used. Carbohydrates are chains of carbon, hydrogen, and oxygen molecules chemically bonded together. When the bond is broken, energy is released, and carbon dioxide and water are given off. So again, remember these leaves are giving off some moisture. The leaves can also give off some carbon dioxide. The lenticels can liberate carbon dioxide, and the lenticels can give off water. So, so trees use this released energy for all biological functions. So respiration, again, this is a constant process of converting food into energy. And yes, this happens even in the winter. So the trees are still consuming because they have to stay alive. Important that photosynthesis food production exceeds respiration, means it needs to make more energy than it's using. When the opposite occurs, that's when trees start to die. When respiration takes place in the absence of photosynthesis, the tree must rely solely on energy reserves. And so think about the winter, or you can think about an insect infestation that eats all the leaves off, or you can think about a very poor pruning job where somebody cuts off all the canopy. And now all of a sudden there's no photosynthetics being produced, so it has to go to that bank of reserves. Over a long period of time, the usage of stored energy reserves will cause the tree to run out of energy and it will die. Oxygen is required for normal aerobic respiration. Trees cannot respire in an anaerobic environment. So think about a flood condition. So a tree whose root system is totally underwater, there's no airspace left in the soil because the water occupies all that pore space. And it can't even go up the trunk and cover the canopy in extreme flooding conditions, which creates that anaerobic environment and respiration ceases. It can also happen in severe soil compaction. So you've seen trees on construction sites that have not only lost roots, but the soils have been highly compacted and the canopies begin to die because they just can't respire. You also have an accumulation of carbon dioxide in the soil, which also, just like us, we can't live in off of carbon dioxide. When oxygen is scarce, respiration uses food at a much higher than normal rate. Sort of think about it like a panic response. It's like, oh, I'm starting to suffocate, and you just start hyperventilating. And eventually, if you were in a low oxygen environment, that would not be good. And so same with the roots, eventually it would lead to root death. So transpiration. So again, we talked a little about it earlier, the transpirational water loss in the form of water vapor is also affected by cuticle thickness, presence of hairs on the leaf surface, number and location of hot, dry conditions might produce a thick cuticle or smaller leaves or sunken stomata. Stomates usually open during the day and close at night. So notice this says usually. So it can occur at any time of the day, but usually they're going to be open during the day and close at night. However, in very high temperatures and high humidity environments, they'll close during the day. And during times of high production after sunset, they can still stay open to have that gas exchange with the surface. Antitranspirants can basically plug the stomates. So if you were going to, say, move a tree in the summertime, you would want to spray it with an antitranspirant so that you can force the trees to close those stomates. But it's not a very common use of the product. I mean, it's not a product that is used commonly. Transpiration can reduce evaporative cooling, uptake of carbon dioxide, and photosynthesis. This should be the use of the antitranspirant. So you don't want to leave these antitranspirants on there for very long because it can cause the tree to overheat. So if you're not allowing the tree to give off water vapor, it's not keeping the leaf cool, so it can overheat. And so also you're going to limit its source of carbon, so the process of photosynthesis can come to a close if you overuse antitranspirants. Water is essential for all living cells. It's sort of the driver for photosynthesis. It's what's going to carry everything. It maintains cell turgidity, and then in other words, keeping that leaf nice and stiff. It's necessary for the transport of essential elements from within the xylem tissue. Water is absorbed with mineral elements from the soil by the roots, and it's used for growth and metabolism. Most water, again, is lost through transpiration. Water loss creates the transpirational pull. Again, it's like sucking on the end of a straw. So as it evaporates off the top, it's pulling the water molecules in through the bottom. A xylem is a continuous column of water. Water enters young roots or mycorrhizal roots by a process called osmosis. So these open cells, these open vessels are not just sticking out there in the soil. There is a cell layer, a barrier there. So it has to move from the soil into the plant across a membrane through a process called osmosis. Osmosis, the movement of water through a membrane from a region of high water potential to a region of low water potential. So in other words, if there's a demand for moisture on one side, it's going to move across the barrier to where it's needed, and it can move either into a plant, it can actually move out of a plant back into the soil. That's important to keep in mind. Pure water has the highest potential. Water with minerals or sugars lower the water potential. Water normally moves into roots where the water potential is lower than in the surrounding soil. If water potential is lower in the soil than in the roots, water will move out of the roots back into the soil. So think about drought conditions. If you have a nice healthy plant in extended drought, well, it's going to begin to take moisture out of the plant and put it back into the soil as the soil demands it. Let's talk a little about this source and sink. So photosynthetes move through the phloem from a source, in other words, like the leaves are the source, that's where it's being manufactured, to a sink where it's going to be used or stored. So if you think about a large oak tree that has a lot of trunk diameter, that's a lot of sink. You have a lot of cells there that have a demand on photosynthetes that are saying, hey, feed me, feed me. So, if you cut off too many leaves on older trees, well, you're going to run into the problem where your sink is going to outpace your source. Leaves are the source of photosynthate. Almost all plant parts are sinks at one time or another. An exception to this might be crepe myrtle. You've seen the green on the bark. Anytime you see green on the bark, even out on small twigs, that photosynthesis can occur in that area also. So crepe myrtles, that's what makes them so bomb-proof, is that they have the ability to produce photosynthetes in places other than just the leaves. Most photosynthate is utilized or stored in proximity to where it's made. So if this leaf is making 100%, well, it takes about 30% to keep those cells going and to keep the cells around it going, and then another 30% down the branch, and then there might be a little bit left over at the end to give to other parts of the tree downstream. So again, here's an example of the source and sink. We have xylem tissue that can move stuff up into the canopy. The phloem, this is where the photosynthetes are going to be. So you might have these companion cells. Here's a diagram of a leaf. So the leaf is producing sugars. These sugars are going to accumulate in the companion cell adjacent to the leaves. And then it can move it into the phloem as demand occurs. So here we have some demand by a companion cell and move it into a sink like a root cell. This large organelle over here is a vacuole. It's sort of an energy or fat storage area for the cells. A little more about transport. Movement of water in the xylem and photosynthate in the phloem are examples of longitudinal or axial transport. Radial transport is the horizontal movement of water or nutrients within the tree between cells of different ages, meaning that it can go towards the center. Remember, xylem cells towards the center are older than the ones out by the bark. So it can the result of the interaction between the genetic potential and environmental conditions have a direct impact on growth and development. So the height of the tree, the color in the fall, the form the tree takes. Plant systems respond to environmental stimuli. Such typical stimuli are light, gravity, and temperature. Gravity is the most constant. The process in trees are controlled in part by plant growth regulators or plant hormones. These are naturally occurring compounds that act in very, very small amounts to regulate things like cell division, elongation, flowering, fruit ripening, leaf drop, dormancy, root development, These are the things that trigger the tree. They don't have a brain, so they have all this chemical stuff floating around that tells things to turn on and turn off as a response to the stimuli of light, gravity, temperature, wind, that sort of thing. These are the bulk of the main plant hormones: auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Auxins are produced primarily in shoot tips. It's also important in the role of root development. Synthetic forms are sold to enhance rooting of cuttings, like rutone, stuff like that. Also, some herbicides can be made out of these auxins that tell the plant to grow as fast as you can, and then they die because they grow too fast. Auxins are also involved in tropisms. This is the orientation of the direction of growth in response to an external stimuli. So geotropism is the upward orientation. Remember, we talked about that acorn that was sending up some seed leaves and sending down a root, a tap root to go check things out. So that's the fact that it knows which way to go is geotropism. Phototropism means it's going to grow towards the light. So if you have trees in a forest and you come up to a lake, the trees, if this is the lake, the trees aren't going to grow like this. They're going to grow kind of like this over the lake because there's no competition for sunlight over the lake like there is out in the forest. So that's a phototropism. Cytokinins are produced in the roots. These are instrumental in root initiation and root growth. Auxins and cytokinins exist in a delicate balance in the plant, regulating shoot and root growth. So the amount of auxin that's produced and the amount of cytokinins that are produced, their relative availability compared to each other, sort of that ratio, is what excites canopy development or excites root development. Growth regulators present in terminal buds inhibit growth and development of lateral buds on the same shoot. Remember we said that the tip's what controls the growth behind it. Strong dominance is confined primarily to the current season's shoot growth, after which lateral buds can start to grow. So the current season might be spring, so it's going to be primarily shoot elongation, and then at the end of spring, beginning of summer, then the rest of it's going to grow some as well. If lateral shoots outgrow the original terminal shoot year after year, well, instead of having this nice pointed Christmas tree, well, it's going to become more rounded, or will be produced, decurrent form will be produced, such as oaks and elms. Excurrent trees have strong apical dominance, resulting in this very upright, like most conifers, are going to grow straight up. Sweet gums and tulip poplars, usually, a nice, straight, tall trunk, single leader, ideally. So, those are example of excurrent trees, but even at maturity, even excurrent trees will start to look a little bit. All trees start off with excurrent traits as juveniles. So again, here's our excurrent tree versus our decurrent form. And then within the same species, here's some young pines. You can see they're growing rapidly, have a very strong central leader. Over here, we have some maturing pines, open grown, and the canopies are nice and rounded. They're not so pointy. Delicate and changing balances, chemical signals that control metabolic processes of photosynthesis, respiration, and biological function. Tree growth regulators impact each other and remain in this sort of dynamic equilibrium. Dynamic meaning that it moves based on stimuli. Environmental stimuli and human stimuli trigger changes in regulator concentrations. Again, if you prune off all the canopy, you're pruning off all the auxins. If you're cutting all the roots, you're cutting off all the cytokinin production. And so it does not stay in balance. Common question that we get all the time, especially in the construction environment, is if we lose a lot of roots in construction, should we go ahead and prune off an equal amount of canopy above ground so that we keep these things in check? And the answer to that is no. Don't do that. We want the tree to get back to that stable dynamic equilibrium. So by leaving one hormone in excess concentration of the other, that's what's going to trigger the formation or accelerate the replacement growth. Defense mechanisms, again, thick bark, thorns, leaf hairs, thick cuticles, cellular membranes that resist decay or ingestion by insects, production of chemicals that restrict insect feeding, pathogen infection or decay. So that gets us sort of to compartmentalization now. So CODIT, coded, compartmentalization of decay in trees, is a model developed by Dr. Alex Shigo. This is just a way to model. It's not a literal process, but it is a model that we use to understand decay. The terminology here is to wall off an injury. So again, we don't use the word heal. We say that it walls off or that it compartmentalizes the decay. Compartmentalization limits the spread of discoloration and limits the spread of decay in the tissues, primarily the xylem tissues. After an injury, reactions are triggered that cause the tree to form boundaries around the wounded area. So remember that cell in the tree is sort of fixed. There's no cellular replacement. So it's likely to have cells above it, cells below it, cells, older cells to the inside of the tree. And so it needs to respond in 3 dimensions. It's not just as simple or straightforward as one little boundary layer. So the coded model helps us to understand the 3-dimensional aspect of compartmentalization. It forms 4 barrier walls. And here they are in order of construction. So wall 1 reverse the vertical or resist the vertical spread of the injury. So wall 1 goes sort of up and down the tree. So if this is my damaged cell, the cell above it is in the cell below it. These are going to be where wall 1 is developed. So I'm going to primarily, that's the vascular tissue. So it's going to have to plug the vascular tissue. An example is like Dutch elm disease, when it gets an infection into the xylem tissue. The xylem says, hey, I don't want this stuff to spread. So it starts producing a substance called tyloses. It plugs the vascular tissue. And of course, if you plug all the vascular tissue, what happens? The tree dies. So wall 1 can be a very vigorous reaction, but let's think about this in more simple terms and think of it as a physical injury. Maybe somebody drives a nail into a tree or something like that. If you have the capacity to introduce decay on that nail, so it's going to want to try to limit it from growing up and down. Wall 2 resists the inward spread by depositing chemicals in the older wood, so towards the inside. So remember, we have these growth increments. So you have the end of the previous year's growth increment. Here's this year's growth. Here's our nail sticking into this year's growth. So the first thing we're going to do is try to prevent the pathogens from being able to get up and down. We're going to try to prevent them from getting into older tissue. So now we have sort of the inward and the upward. In wall 3, resist lateral. So the sort of the 3rd dimension here is growing around the trunk of the tree. So we have those ray cells. Do you remember those? And I said they played a big part. This is where they come into play. So eventually you're going to have what looks like a truncated pizza. So you're going to have this sort of area that comes down in a V shape, and then in the back end of it, or the inside of it, if we turn it this way, is going to be last year's growth increment. Here's a ray cell, here's a ray cell, and then here's the outer part of the tree where the bark was. So the outer part is what we call the outer reaction, is what that's called wall 4. And it protects from, this is a physical barrier. This is not a chemical barrier, because remember, you damage the cells to the outside. So it's going to have to grow new cells around where the damage was. Doesn't replace the old damage. It just grows around it and incorporates it or closes off. So here we have another look. So again, this is looking down on top of cross section. You can see where the ray cells, this was a ray cell, this was a ray cell. That's where the lateral spread was stopped. This brown, you can see this sort of light colored ring that goes all the way around. That's where wall 4 started. So this was the exterior of the wound at the time the wound occurred. The distance from here to here is the amount of growth that has occurred since the injury. That's wall 4. So that's that nice physical wall. And here's an example of a pruning injury. And so the walls kept failing. And so the limit of decay just kept going down the trunk. Here's a trunk injection, like when you drill into the tree and put in a little chemical. So the injection created this small wound inside of the tree that went up, but eventually was stopped going up, and it was stopped going down. And notice all this white growth on the outside after the injection was made. So this is wall 4. Here's some more just examples of the model. Wall 4, the damaged area was so big that it's going to have trouble closing over the wound, so it might lead to some pretty big hollow on the inside. This is what we would like to see happen, where it's totally enclosed and all the growth since wall 4 is nice and clear tissue. You can see that wall 3 began to fail here, the lateral wall along the ray cells, and eventually it has failed all the way around. So, but it held tight for a long time and then rotted out. And then the rest of it gave up the ghost. So eventually this is going to become a perfectly hollow tree. When walls 1, 2, and 3 fail, decay can spread inside of the tree. A hollow cavity is formed. The barrier wall 4 is strong chemically, but is weak structurally because the structural role of the parenchyma rays is discontinuous at wall 4. This is how you need to look at it for the exam. We now understand that wall 4's structural capabilities are much stronger than we originally thought, now that we've done so much risk assessment and analysis, but this kind of persists. So it's not really as strong resisting additional decay because the vascular system's a little disrupted and you don't have the ray cells that protrude into the early stages of wall 4. The process that resists the spread of decay into new growth can also lead to shakes, lengthwise separation between growth rings and cracks. So what that's saying is that as you have this very dense and somewhat drier material forms, that wall 4 forms, it grew at a different rate than the inside, than the regular xylem did. So as the tree flexes and does things, you're likely to have separation at that wall. And then now that brings us to tropical trees. So anatomy varies greatly, more so than that of temperate species. So the way that tropical trees look can be very different from one another. They may totally lack annual growth rings because growth can be more or less continuous, or may be highly regulated by wet and dry seasons. So think about the tropics and the rainforests and things that occur at the equator. We also have deserts on the equator, right? So we don't only have rainforests there. So we have difference in species that occur, even though we have relatively the same amount of daylight, spring, summer, winter, and fall. So because of that, they're not really going to experience dormancy. However, there's probably a few that do. They may have extremely large foliage. So tropical trees or palms have some of the largest leaves out there. Some of the other deciduous trees, or not deciduous trees, but other hardwood trees of the rainforest can also have just enormous leaves, trying to get as much light as they can. So some have very wide spreading buttress roots because the soils down there are not very thick, and the roots tend to grow right on the surface. So these buttress roots can be enormous, but are necessary to help to distribute the mechanical loads onto the soil, keep it standing upright. Some species even produce aerial roots. These are, if you have a branch and then the root can drop down through the sky, through the air, down until it hits the soil. And when it hits the soil, it can root again and start absorbing nutrients. And so these are called aerial roots. They can be used for gas exchange and propagation. So once that aerial root drops roots into the ground, you could cut it off here, and then you have sort of a genetic clone of the parent plant. New plants that are propagated in this manner are called clones. Rapid growth rate due to favorable growing conditions, so they can grow quite rapidly. They transplant easy. Wound closure and regrowth after injury can be rapid. However, when these trees are removed from their native ecology, it becomes a challenge and they don't behave the same way. So like our house plants back here behind me wouldn't function the same if I planted them outside as they would inside. So I have to sort of mimic their growing conditions inside that they would have found in their native ecology. Now let's talk a little about palms. Palms are monocots, as Kay told us earlier. And they are biologically similar to grasses. It's not really a grass, but they are biologically similar. Do not have a cambium layer or growth rings of xylem. So here's a cross section of a monocot here, of a palm, and we have these vascular bundles. Remember, inside of the leaf, we talked about the vascular bundle. These palms also have vascular bundles evenly distributed throughout their trunk. These vascular bundles, however, are surrounded by a very fibrous sheath, and they are embedded in a matrix of all this fiber, and that's what allows them to stand up and grow tall. These fibers are made of parenchyma cells. Stem develops all at the vascular bundles. It's going to grow all of them that it needs behind that primary bud as it grows. So it's going to bring along all that tissue as the thing gets taller and taller. Palms go through what is referred to as an establishment period. So when they first emerge, when they put that first single leaf out, it begins to produce photosynthesis, it begins to produce energy, and it gets wider and wider and wider as it grows more and more of these vascular bundles. But it doesn't grow in any height, it just grows outward. It might get a little taller, but not a lot. It'll put on a few more leaves and that sort of thing. And it might sit in this establishment period for years. At some point, it reaches whatever its genetic optimum is for its size, given its location, and then it begins to shoot up. So once they shoot up, that means it's attained their maximum girth. Vascular bundles retain their conductivity throughout the life of the plant. So those very center bundles are just as alive as the ones that are out closer to the surface. The stem is considered woody, although it's different anatomically from the wood of hardwood and conifer trees. Cells in the stem thicken and strengthen as they age, so it's continually putting on some of this fiber. The stem is capable of storing starch in these parenchyma cells, so it still has a big storage area. Movement of water and minerals is not restricted just to the outside, so it can, if the trunk gets some sort of damage, the interior is still very functional. Again, there's no secondary growth on palms, not have the ability to compartmentalize because they don't have that secondary growth. So wounds to a palm are permanent. They aren't closed over. They aren't compartmentalized. So decay is forever. I mean, the injury is forever on the palm. You can forever see it. Resistance to decay, what gives them their ability not to just sort of decay and fall over, is this hard sheeting fibers of the vascular bundles. Typically, there's only one apical meristem per stem. Primary bud is protected by the overlapping leaf bases of the emerging fronds. So remember, they kind of come out like a fountain. So from the center outward. If the bud at any point is lost, the entire stem will eventually die. This is a common question after tornadoes and hurricanes. It's like it ripped the sort of the center. I still have leaves all over my palm, but the little center part is gone. Is my palm going to make it? And the answer is no. Eventually, it does run out of energy because it's not producing new leaves, and it does die. Photosynthesis takes place in the fronds. Fronds consist of a blade, a petiole, and a leaf base. Leaves are produced sequentially. Again, so the newest leaves are in the middle, the oldest leaves are to the outside or the base. Palms have the largest leaves of any tree. Flowers and fruit emerge in or below the crown from the leaf axis, the upper angle between the leaf and the stem. Leaf scars or the node area, sort of the petiole area. Flowers are clustered in large aggregates of many flowers. Flowers, or inflorescences, can be inconspicuous or very showy. Some palms flower once in their entire lives and die, and others produce an excessive amount of fruit, such as date palms, things like that. The palms can be, you can have tons of it, like date palms, or you might only have a few, like coconut palms or something like that. The root system of palms are very different from those of hardwood trees. Again, they're going to stay that same diameter from the time they're produced all the way out several times the height of the plant in all directions. So palm roots, again, they lack secondary growth because there's no cambium there. Roots other than the primary seedling roots are adventitious, so they just come about here off of the trunk. You can see there's just thousands of them. They develop at the base of the stem during root initiation zone, in the root initiation zone, which is in the upper 12 to 18 inches of soil. So all this fibrous root development could go down as deep as 18 inches. They grow very close to the stem in a densely packed network called the root mat. So the closer you get to the trunk, the more mat-like in appearance the roots become. The further away you get, the more sort of open they are. If you cut these roots, they do re-sprout. So if you've ever seen a palm tree like on to Las Vegas and seen them shipping in these palms that are 100 feet tall and have a root ball on them that are about this big around, it's because they don't need those roots to regenerate new roots. Once you plant that thing in the soil, that the hormones are going to trigger the growth and development of the new root system, and it will regrow all of this stuff really rapidly. And palms do form mycorrhizal associations. All right,